In today's tutorial, we will talk about the beta oxidation, and we will discuss some very important points regarding the beta oxidation. Now, these points will help us understand this topic in a very easy way. Now, what are those points? The very first point is oxidation. Second point is types of oxidation, beta oxidation, the site where the beta oxidation occurs, and the steps of the beta oxidation. And the last point is the beta oxidation, enzymes, coenzymes, prosthetic groups. And here I will tell you people about how to remember the enzymes involved in the beta oxidation. So let's start from the very first point, oxidation. If you remember this oxidation and if you learn about the oxidation, then it will be very easy for you guys to understand what actually beta oxidation is. So what is oxidation? We have a very big discussion regarding the oxidation and reduction, means regarding redox reactions. So well, here I will just tell you people in short that oxidation is the addition of oxygen, loss of hydrogen, electron, ion. Now this is becoming again a very big definition. Just to make it short further, consider oxidation is the loss of proton. Just remember this. Oxidation is the loss of proton. Remember this particular definition for this particular lecture. Regarding the beta oxidation. Then it will be very easy for you guys to understand the beta oxidation in a very easy way. So now let's come towards the next point, that is the types of oxidation. We do have different types of oxidation that are the alpha, the beta, the omega, and etc. Means we have different types of oxidation. We have oxidation observed in the saturated, unsaturated fats, etc. Means we have different types of oxidations. So now what is the beta oxidation in reality? Now let us understand it in a very easy way. The very first thing that I want to make you people understand about is beta. What is beta? And then what is oxidation? We got the concept of oxidation. Now let's come towards the beta. Here we have the fatty acid. Now in this fatty acid, regarding chemistry point of view, we have certain names for each carbon. The carbon just after the carbonyl functional group here in this fatty acid this is given the name as alpha carbon, and this particular carbon is given the name as beta carbon. And we have number of carbons available here that I have enclosed in this R. So when if you write R here, it means we have number of carbons available here. Well, in order to bring our discussion to the very specific point, that is the beta oxidation, I'm just skipping other positions of the carbons, means other names of the carbons. Now consider this fatty acid structure for the beta oxidation. So now what is beta oxidation? It is actually the oxidation of the fatty acid at the beta carbon. Now here is our fatty acid. So what happens if there is oxidation at this beta carbon? Then it is known as beta oxidation. Super easy. Oxidation at the beta carbon of the fatty acid is called beta oxidation. Simple, clear, easy. We cleared that oxidation is the removal of proton. So if you remove the protons from this beta carbon of this fatty acid, this is actually known as beta oxidation. Now here we have a carbon, and with this carbon we have two hydrogens, and here we have the carbon, again here are the two hydrogens available. So if we remove the hydrogens from here, that is called as oxidation, and actually if we are removing from this beta carbon, that's why it is known as beta oxidation. I think it is super clear, super easy. Well, if still you don't understand, you can ask us in the comment box. Now let's come towards our next point. That is the site of this beta oxidation. The very important site is the mitochondria. The beta oxidation actually occurs in the mitochondria, within the mitochondria. Now let's come towards the next point, that is the steps of the beta oxidation. What are the steps involved to accomplish the beta oxidation? The steps are activation, transport, beta oxidation. Now these three steps, these three mechanisms are supposed to happen with the fatty acid. Our fatty acid is supposed to activate, and now this first step, activation, is actually done in the cytosol, and transport is done from the cytosol into the mitochondria. And we told you that beta oxidation occurs within the mitochondria, means inside the mitochondria. So these are the steps. So now let's start from the very first step, activation of the fatty acid. The very first step, and here we have the fatty acid. Remember guys that the activation is actually occurring in the cytosol, and then from the cytosol these activated fatty acid will then move towards the mitochondria. Well, so now how this fatty acid will get activated? Here we have our CH2 double bond OH, our simple fatty acid. Now how this fatty acid will be activated? Very simple, by the application of an enzyme known as thioester and ATP. ATP will act here. By means of the usage of ATP, we will use ATP here, means ATP will be used here and an enzyme will be used. then our this fatty acid will be activated, then it will be known as acyl-CoA. Now this acyl-CoA, it is supposed to be transported into the mitochondria, the very second step, transport. This transport will be done by using a car. I'm just telling you a car because this is actually having a pronunciation matching with a car, known as carnitine. This carnitine will actually take this acyl-CoA and will do the transport of this acyl-CoA from the cytosol into the mitochondria. So like this, it will bring it in the mitochondria. And here again, an enzyme is involved, known as carnitine acyl transferase, CAT, type 1. Here, when it is transported into the mitochondria, again, an enzyme known as CAT type 2, carnitine acyl transferase type 2 is involved, which is involved for the separation of these two components, carnitine and acyl-CoA. Now this our acyl-CoA is available for the beta oxidation. And remember, for the beta oxidation, we do have further 4 steps: oxidation, hydration, oxidation, cleavage. Oxidation is repeated, okay? Once oxidation, then hydration, then again oxidation. And then we have the last one, that is the cleavage. So what will happen in the beginning here? Now this RCH2, CH2, C double bond O, COA, acyl-CoA is available here for the beta oxidation. Now we have learned that what is beta oxidation? Actually, the removal of the hydrogens from the carbon, the beta carbon, is actually known as oxidation. Now here is our alpha carbon, and here is our beta carbon. Remember, alpha carbon is the carbon just after the carbonyl, this one, functional group, okay? So now here at the beta position, there will be oxidation. The very first step of the beta oxidation is actually the oxidation. Now this oxidation is done by an enzyme known as dehydrogenase, because hydrogens are going to be removed from here, and these hydrogens will be taken by a prostatic group, this one, known as FAD, flavin adenine dinucleotide, and it will become as FADH2. Why? Because 2 hydrogens will be removed. These 2 hydrogens will be removed, then these 2 bonds are available to interact. Like this, a double bond will be produced, and 2 hydrogens are removed by using an enzyme and a prostatic group. In the very first step, what happened in the oxidation? We used enzyme and a prostatic group. RCH double bond CH and bond double C double bond O, COA is formed, and now this newly formed product is having enol in its name. Well, now this enol will be treated with the water. Second step is hydration. Here again an enzyme will be used, known as hydratase enzyme. This enzyme will actually add the hydrogen and hydroxyl ions of the water into this particular compound. So now you know water is formed by the combination of hydrogen and hydroxyl. When these two react together, they will make water, and when you break water, again these two will be formed. Now if we add these back, what will happen? So we will add actually hydroxyl to this beta carbon, and the hydrogen will be added to the alpha carbon. And after adding the hydrogen and hydroxyl, this will be the product you formed: RCOH of the water, H of the hydrogen, and like this the product formed will be having the hydroxyl hydrogen in its structure. And the next step that is going to happen is the oxidation again. Now here again an enzyme will be used and a coenzyme will be used, CE, a coenzyme known as NAD, and this NAD will be converted into NADH2. Why? Because the enzyme used here, known as dehydrogenase, will remove the hydrogens, 2 hydrogens, and those 2 hydrogens will be carried by the NAD, and it will produce NADH2. So here we have 1 hydrogen, here we have another hydrogen. So these 2 hydrogens will be removed. What will be formed then? RC double bond O. Why? Because this bond will become free then, because hydrogen is removed, and again another hydrogen is removed from here. Then this bond with free here will come and will bind with this oxygen, and like this the valency of oxygen will fulfill, and the product formed will be like RC double bond O, CH2, C double bond O, COA. Now this product is supposed to undergo the last step, that is the cleavage step. Here again an enzyme will be used, known as thiolase. This thiolase will split this particular product into 2 components. The first component is CH3, C double bond O, COA, known as acetyl-CoA, and the second one is acyl-CoA, RC double bond O. Now the bond is actually going to break here. Here the CoA will join this RC double bond O, known as acyl-CoA. Now this acyl-CoA is supposed to again undergo the process of beta oxidation, and here a product of the fragment known as acetyl-CoA, which is a true carbon chain, will be separated, and then this acetyl-CoA is supposed to enter the Krebs cycle, and then again further energy will be produced. So like this, this remained portion of the acyl-CoA will again undergo the process of the beta oxidation. And this fragment is supposed to undergo the beta oxidation time and again until this is completely oxidized. Suppose here we have 30 carbon long chain. Now this 30 carbon long chain of acyl-CoA, this will produce, now divide this 30 by 2, you will have 15. This will produce 15 acetyl-CoA. What? 15 acetyl-CoA. This acyl-CoA will produce 15 acetyl-CoA, and then this 15 acetyl-CoA will be produced actually by repeating this acyl-CoA fragment how many times? 14 times. Just subtract 1 from the acetyl-CoA, you will get the number. Now how will you understand about this particular strategy? Remember, the longer the chain is, just take the chain, divide by 2, then you will get the number of the acetyl-CoA fragments produced, and then just minus 1 from that fragments produced, you will get the repetition or the cycles for this acyl-CoA fragment in order to complete or in order to provide these particular acetyl-CoA fragments. So if we have, suppose again, another example, if you have 20 carbon long chain of this acyl-CoA, so it will produce how many acetyl-CoA? This will produce about, now divide by 2, you will get 10. This will produce 10 acetyl-CoA by repeating this fragment how many times? 9 times. Now you got the concept. Chain divided by 2, you will get the number of acetyl-CoA, and minus 1 from this 10, you will get the time or the repetition this acyl-CoA is supposed to undergo in order to do the oxidation or to produce the required acetyl-CoA for the Krebs cycle. And you know the Krebs cycle uses or utilizes this acetyl-CoA in order to produce further energy. Now let's come to the next point, that is the beta oxidation. The enzymes involved, the coenzyme and the prostatic. Now if you want to remember that what are the enzymes involved, what are the names of the enzymes, and what are the prostatic groups involved, you don't need to be confused regarding this point of view. Just remember the structure, remember the steps, and then from the same steps you can gain what are the enzymes, what are the prostatic group. Remember, whenever there is oxidation, there will be the usage of enzyme. With the enzyme, there will be the use of either prostatic group or the coenzyme. So in the first step, we used the prostatic group known as FAD, and we used an enzyme known as dehydrogenase. You know, when there is oxidation, there is removal of the hydrogens. So when there is removal of hydrogen, it is obvious. So hydrogen will be removed by the enzymes which are known as dehydrogenase. So you can guess the names yourself. Dehydrogenase is actually removing the hydrogen. So you just write dehydrogenase, your point will become valid then. So what is then hydration? You know the enzymes responsible to do the hydration are the hydratase. So it is easy now to write the name of the enzymes involved in this particular beta oxidation. And again, oxidation, remember the steps. When there is oxidation, enzyme is actually responsible to remove the hydrogen. So what will be that kind of enzyme? What will be the name of that enzyme? Very simple. The name will be dehydrogenase enzyme. So dehydrogenase is actually removing the hydrogens, and that is responsible in the oxidation reaction. So now here oxidation is going to happen by means of the dehydrogenase, and those hydrogens removed will be taken by the NAD, which is a coenzyme. So first we use a prostatic group, then we used a coenzyme. Prostatic group was FAD and coenzyme is NAD. So FAD was converted into FADH2, and NAD is supposed to convert into NADH2 because the hydrogens are supposed to be the part of these NAD and FAD. And the last, we have cleavage, and you are supposed to remember the name of this cleavage enzyme, that is the thiolase. And that's it, very simple understanding regarding the particular topic, that is the beta oxidation. And if still you have confusion in any point, you are free to ask time and again in the comment box. We'll come for the answers very soon. Thank you for watching.